share screen. So I think I could ask them to join. So whatever we have learned yesterday, are you all comfortable with it? Any questions? No? Okay. What I'll do is I'll just give it a brief recap. So this is going to be the last class on Dynein structure as such. I'm not going to go into details of all the associated proteins. As I mentioned in the very first class, Dynein itself is a huge polypeptide, right? It's like 500 amino acid plus. Sorry, 500 kilodalton plus. And associated proteins and etc. take the total molecular weight of this huge complex even higher. And for it to function, I don't see why they are not, I can't see why they are not able to join. I see they are online. Anyway, so for it to function, there are other complexes with which it interacts, such as dynactin, and dynactin itself is as huge as dynein. So I'm not going to go into the nitty-gritties of all of these complexes. The idea is to just give you a background about the motor protein itself and how it works and how it functions. So we will carry that forward today. I hope you were all able to join without any glitches. Because I forgot to schedule the meeting. It was okay for you all to join?
Yes,
Yes, ma'am.
Yes.
Yes.
Okay, then I will leave. Let the others join themselves. Okay, so we come back to the same old slide. What do we see in the slide? We see this huge polypeptide chain with an N-terminus. And the N-terminus tail, as I said the last time, it's associated with a number of different proteins. And these proteins function to modulate its activity, modulates its cargo binding activity, what kind of cargo it will bind to, what kind of cargo it will select and carry into the cell is going to be determined by the interaction of the associated proteins with this tail. And this is the region that is most mysterious because we do not quite know the structure and how it is present inside the protein. So you see everything else, you can see this is like a coiled coil. Here you see beautiful structures, but for this we do not quite know. So it's kind of obscure even now. After that you have something called the linker, and we will talk about the linker a little today. And then you have this AAA plus ATPase kind of domains, and you subdomains, and you have this 6 domains that are present one after the other. And between these domains, you have a region called the stalk. So this stalk has a microtubule binding site at its end, and the stalk actually forms like a coiled coil. So as a coiled coil structures. So coiled coil, if you remember, coiled coil we had also seen in myosins, where myosin tails can actually form these coiled coils. So there's one helix, there's another helix, and the two helix wrap around each other, you form a coiled coil. So you have this coiled coil structure, and you have something called buttress. We'll come to this, and followed by you have C-terminus. So this little region is the minimum motor unit that is required for the dynein to walk on the microtubule, as well as to have a sliding motion of the microtubule. This tail is, or the N-terminus is mostly required for cargo binding as well as for dimerization activities. forms dimers, and the dimers walk on microtubules. तो ये हमने last time देखा था. So last time we had asked the question that if there are six AAA plus domains, then which of these six will actually be hydrolyzing ATP? And in the very first lecture, I had said that it's the first AAA1, which is primarily responsible for ATP hydrolysis, and therefore the movement of the entire dynein protein is dependent on it. Yesterday we found how exactly this was concluded. So the experiments they did was firstly based on understanding the minimum structure and sequence that is required to bind to ATP. And they found that it is present in these first 4 AAA plus domains, not in the last two, right? Last two many. And then there is a very specific sequence that is repeated, very nearly the same. You see there are some small modifications, but nearly the same. And always there is a lysine, and we know lysine is positively charged. ATP has a huge negative charge. So if you can disrupt this positively charged lysine, put in something else there, which doesn't have this kind of a charge, then very likely you're going to affect ATP binding, ATP hydrolysis, and that's what they tried. They mutated this lysine residue in each of these individual 4 AAA plus domains. And what we saw last time was that अगर आप wild type का मतलब क्या है? What is wild type? Wild type is the gene as it is. जैसे आप dynein dictyostelium में देख रहे हैं या human cells में देख रहे हैं या mouse में देख रहे हैं, जो gene sequence exactly उनमें है, that is the wild type. And if you alter the gene sequence, if you mutate it, then that is a mutant, right? So wild type, mutant. तो wild type जो sequence है, wild type sequence का अगर आप dynein लेते हैं, and it's a truncated dynein, right? Last time हमने ये भी देखा था, ये पूरा dynein नहीं है, सिर्फ minimum motor unit. उसका मतलब है the N terminus that is required for cargo binding is taken away. So if you take now the minimum motor unit and you try to do a microtubule binding assay, this is what they did. They took microtubules, they took these dyneins, mixed them in the presence or absence of ATP, and then tried to see how much of it is associated with the microtubule, how much of dynein, and how much of it is free. So in the absence of ATP, most of the dynein wild type is associated with microtubule. And so is the case with all the mutants of the AAA domains. But जब हम ATP डालते हैं, the moment you add ATP into wild type, you see this shift from the polymerized tubulin to solution, which means that if you have tubulin, if you have these dyneins walking on microtubules, the moment you add ATP, these dyneins sort of detach from microtubules. So there is a sort of detachment, and that is evident here. You add ATP, the dynein detaches from the microtubule until there is a hydrolysis, and that creates a condition where it again seeks out the next binding site of microtubule and binds to it. So this is clear. But if you now look at the mutant P1, that is AAA1 and AAA3, then you see this is not the case for these two mutants. So these two mutants specifically have a problem that in presence of ATP, they still seem to be tightly bound to the microtubule, which indicates that these the AAA1 and 3 are actually the points where the ATP is going to be binding, and maybe there is a problem with ATP binding itself, so it's not attaching. Second, they found was that ATP hydrolysis itself wasn't occurring in these two domains. So here you see ATP hydrolysis rate almost with time, with concentrations. You see there's no change, whereas this is wild type. With 3, there is some amount of ATP hydrolysis, very little, but there is some. And they found that if you mutate AA1, you can definitely stop the sliding movement of microtubules. But for AA3, you still retain a bit, like partially you retain the sliding movement. All of these goes to show one thing, that AAA1 subdomain is the most important domain for ATP binding and hydrolysis. So the entire power generation for Dynein motion occurs through AAA1. So इतना तो हमने देखा था. And what we're going to come to now is so let's kind of recap what we know about how Dynein works on microtubules. So microtubules, microtubule Dynein interaction, as I've said before, you kind of think of it as an analogous to myosin binding to actin. So similar to that situation here, if you have, the moment you have ATP bound to the pocket in the AAA1 domain, the Dynein motor binding domain detaches from microtubule. So it's no longer attached, it kind of detaches. And then simultaneously, it tries to find the next binding site. Now, here is a catch. Here is a small catch. It is not quite clear, at least I haven't seen it, and maybe it's not very clear yet, whether the ATP hydrolysis in the AAA1 domain is actually occurs before it binds to the next site, even loosely, and maybe that loose attachment sort of enhances its ATPase activity, or whether it's a spontaneous ATPase activity and then goes and searches for the next. I mean, which of it is first and second? Maybe both of them occur. I'm not very clear about this, but whatever it is, you have ATP bound to the pocket, and it's very clear that that has a completely different conformation, and it doesn't bind to microtubule. But when ATP is hydrolyzed to ADP and PI, that has a strong attachment on the microtubule, very strong attachment. It binds strongly, and then release of the PI creates a power stroke. And this power stroke can be, the whole thing can be sort of brought back to normal when the ADP is released and you have an empty pocket, and then ATP binds to it and then detaches. So if you go back to this slide, you see the moment you add ATP, you are seeing a release of these dyneins from microtubules. This is the majority of microtubule with very little of dynein, but most of the dynein is actually released because you're bound to ATP. So this, what I would like to emphasize here is that there is also a change in the way this linker domain fits on the dynein structure as this kind of nucleotide that is bound to the Dynein AA1 domain changes. So in each of the cycle, you can see the position of this linker domain and the position of the stalk keeps changing. So this sort of kind of is a mechanical way of creating that motion, creating that changed conformations so that the dynein actually moves forward. We will look at it in greater detail soon. So before I go, so this is where today's class starts. So before I go any further, I would like to introduce you all to a technique called TURF. And the reason I would introduce you to this is that even previously we have seen that जब kinesins पढ़ रहे थे या फिर जब हम microtubules या actin पढ़ रहे थे, we have seen multiple cases where they have taken a slide and on that they have added the microtubules, so the microtubules are present on the slide, and then perhaps they have watched in real time the motion of kinesin on the microtubules. We have seen experiments like this. But what kind of microscopic technique they have used to visualize this? That's important for us to understand a little bit. So we are going to look at what TURF microscopy is. But before that, I'd like to ask, Does anybody know what turf is? And just for me to understand how much you're aware of.
Yes, ma'am. We know that we have learned in class 10th and 12th, etc. In physics, yes.
Perfect. Both of.
What, ma'am? I did not understand.
Tell us what you know about turf.
Yes, ma'am. It happens when the angle of reflex incidence is more than that critical angles, then that light is completely reflected inside when it goes from a denser medium to what I can say, a medium which have a low refractive index. So in that case, it happens.
Perfect. So Ratnesh has perfectly explained what is total internal reflection, and total internal reflection is something that we are all aware of. I don't know आप लोगों ने किया है या नहीं. हम लोग जब बच्चे थे हम लोग kaleidoscope जैसी चीजें लेके खेलते थे या फिर पेरिस्कोप बनाने की कोशिश करते थे क्लासेस में. So it all comes back to this idea, basic physics idea of total internal reflection. So total internal reflection क्या है? So if you have, so this entire thing I've actually taken up from Wikipedia because I found this image so beautiful. So what is total internal reflection? Total internal reflection, as Ratnesh has said, is that you have, suppose you have a glass and air interface. So glass has a certain refractive index, air has a certain refractive index, right? And because of the change in refractive index, when you have an incident light falling on the interface between these two materials, your light is, the first thing is not total internal reflection. First thing is it will be refracted, right? So जैसे जिस एंगल से लाइट आ रहा है उस एंगल से वो दूसरे मीडियम में नहीं जाएगा. It will be shifted. So if you take a coin, it's a very classic thing. You might have all seen it, done it yourself. You take a coin, एक कांच का ग्लास ले लीजिए, उसमें पानी भर दीजिए. If you now look, you might feel that the coin is actually much higher in elevation. It's all because of the phenomena of refraction, that it's kind of a bending of light, that you could say. So as it moves from one medium to another, you find that there is a bending of light. Why does this happen?
And because of the speed of light changes in different medium.
Yes, thank you. So this is so because of this you end up seeing you have a change of, so that in the two media you will find that the speed of light varies, and then you see that there is a change in the in the what should I say? It's not a direction, but rather it's the angle. So the angle at which it is incident on this the boundary between the two media, and the angle at which it will be moving through the second media is going to be different. Now, if you keep, so this is the incident angle, right? If you keep moving the incident angle, if you keep moving it, you would see that there is an angle called the critical angle at which the light would be grazing between these two surfaces. So that's the grazing incidence. Now, if you move your angle of incidence even further, more oblique, you will now find that this incident light is no longer moving through the second media, but is being totally internally reflected. It comes back, so you have the light moving this way and then it comes back, which means that it is no longer going into the second medium. But how do you then build a microscope based on total internal reflection? I mean, total internal reflection, we use mirrors in microscope. Mirrors would be working this way. But then how, but this total internal reflection microscopy is actually using this whole idea to excite fluorophores. So how would you do that, or why, or how can you do that, or why would you do that? That's the question. Now, when there is total internal reflection, whatever energy you're sending here is coming back, but you have at the point where this incident light hits the boundary between the two surfaces, you have something called an evanescent wave. Now, evanescent wave, if you would like to read any further about it, I would suggest please go online and search about it. You would find some physics explanations about evanescent wave. But for the moment, let's think of it this way: that when you have an incident light which is falling at the interface between these two media, and the two media have distinctly different refractive indices, and you have the light coming from a higher refractive index media, and it's falling at the interface between the higher and the lower refractive index media. So say this is glass, this side, and this is air, and it's falling at an angle that is greater than critical angle, and it gets totally internally reflected. At this point, it is creating something called evanescent waves. These evanescent waves, they um, their sort of intensity falls down exponentially as you go from this point of how do I say? This is the boundary between the two materials. So from this boundary, if you start moving towards the air boundary now, the evanescent wave intensity falls exponentially. So because of this exponential fall, you will be only able to use this evanescent wave to excite molecules which are very close to the boundary. So I will repeat this once more. You have a total internal reflection phenomenon occurring. So whatever incident light you're sending, it's going back into the same medium. But at the point where it hits the boundary between this glass and air, you have something called an evanescent wave generated. And this evanescent wave, the property of this evanescent wave is that it falls or decays exponentially as you move into the second medium, the lower refractive index medium. So this means that you can use the evanescent wave to specifically excite fluorophores which are very close to the surface, not far, because this does not penetrate into the media, second media too much. It's just very close to the boundary, so very thin layer. Now, if this is the critical angle, then as you make your incident light more and more oblique, you will find that the depth of this evanescent wave keeps decreasing. The more it is closer to the critical angle, the depth of this evanescent wave would be higher. So one thing is very clear from this, that okay, let's go here. So one thing is very clear, that you could use this evanescent wave to excite fluorophores which are very close to the surface. Now, before I go any further, I have actually never spoken about fluorescence itself. So can somebody explain here, what is fluorescence? What is happening exactly during fluorescence? So what is fluorescence? हम कहते हैं इंसिडेंट लाइट, then there's an emission, excitation, ये सब है क्या? थोड़ा कोई एक्सप्लेन कर सकता है प्लीज?
Ma'am, I don't know completely, but I think like there are compounds which are molecules which are, when light falls on that, it gives certain kinds of a color, and that is what it is, and we can see that.
No, not exactly, Ratnesh, but we'll come to this. We'll come to this. Who else can try?
Ma'am, can I?
Yes, go ahead, Pranav.
Ma'am, so when molecule absorb light of certain wavelength, then the electron in its shell will go into higher state. So when that higher state will come to the rest afterwards, then after rest, the energy released in that process will be equal to the certain wavelength that is lesser than the incident wavelength, and that wavelength will give a particular color to this reaction. That is called fluorescence.
So can you repeat what you just said? Wait, what I will do is I will stop sharing the screen for a while because I need to use the, I did not think of this when I was making it. I need to use the, क्या कहते हैं उसे? Paint.
Uh,
Repeat it, Farman. Go ahead, repeat what you
okay,
just
ma'am. Sure, ma'am. So when ma'am compound absorbs certain wavelength of light,
एक मिनट
uh,
wait कीजिए
okay,
paint. Yeah, you can see this, right? So
yes, ma'am.
all right, a compound is absorbing certain wavelength of light.
Yes, ma'am.
Okay.
Certainly chromophore, which is having double bond or a pi bond in it.
And
When ma'am it will absorb light, the electrons in outer shell will go into the higher energy state. When they will go in higher energy state, then afterwards they will come to rest. So at process of coming to
higher
rest,
energy state के बाद rest.
yes, ma'am.
So it will stay at the high energy state. How long?
Until the light will be there, incident light will be there.
Until?
Until the incident light will be there.
Until the incident light will be there.
Yes, ma'am.
Ma'am, for a period of nanoseconds.
It, well, I don't exactly remember nanoseconds ki picoseconds, but it will be very, very short-lived. Very short-lived. Very difficult to catch this in this excited state.
Okay.
So, okay, let me get to this. So, the whole idea of fluorescence, fluorescence में है क्या? So what Ratnesh said is that you're shining light on something and then you see that it's of a certain color. You know, this is what he said. And so ये तो फूल पत्ते भी उनके ऊपर जब आप लाइट डालते हैं, so they appear a certain color. Is that fluorescence? No, right? It's not fluorescence. What it means that if you have a leaf, then you're looking at the light that is sort of reflected off the surface and is no longer absorbed. For example, अगर आपके पास, if you go into, so पुराने ज़माने में होता था ना कि photographs एक photographic room में develop किए जाते थे using films and everything, and they used to have a red light. तो अगर उस रूम में आप red कोई चीज़ ले जाते हैं तो आपको वो white दिखेगा because it's going to be reflecting back the red wavelength and absorbing the rest of it. So यहाँ पे आप वो नहीं हो रहा है, you just see it as white. So what I'm trying to come to is that fluorescence is a very specific phenomenon where you have a very specific wavelength, maybe not a very narrow range, but a broad range. So let me go back here. So you can have something like called the excitation spectrum. इसका मतलब क्या है? You have a certain range of wavelengths. So this is the range of wavelength. This on the y-axis is the intensity, and on the x-axis is the wavelength. So you have a certain range of wavelength which the molecule can absorb, and the electron in this molecule can go to something called an excited state. Just as Farman has stated, it can go to an excited state. But this excited state is very short-lived. This excited state is very short-lived. The electron doesn't like to be in that excited state. It tries to come back to the ground state. it tries to come back to the ground state. That's the first thing. This is just exactly what you would think of as absorption. So you have a certain wavelength of light that the molecule, the electron in the molecule can be excited with, and then this wavelength of light causes the electron to go into an excited state, but the excited state is not a long-lived state. It's a very short-lived state, and the electron tries to come back to the ground state. But what happens is that the electron, if it moves from this state, so suppose, let me draw something like this. If it moves from this ground state to this excited state, and it's trying to come back to the same ground state, right? It's trying to come back to the same ground state. And what I'm drawing by these lines are actually energy levels. So it's gone to a high energy level, but it's trying to stabilize itself. It's trying to come back to the lower energy level. So it comes back, but it does not come back to the same state as it was before. So the amount of energy that you need, so I will represent that as nu, which is the frequency, or let's say the energy as E, that you require to push it into the high energy state. When it comes back, the energy that it releases, because it's coming back to a ground state, this energy, say now E prime and nu prime, is not the same. There is a difference because it's not come back to the same state. So if now you look, so if it has not come back to the same state, but it would always like to come back to the same ground state, what it does is the rest of the energy is released as vibration, but here it releases a photon. It releases one photon. How do I write it? My goodness. यहाँ पे एक photon release हो रहा है. So you have, let me draw it once more, energy states. So you have a lower energy state transitioning to a higher energy state. And you have excited this molecule with a certain wavelength, which corresponds to a certain frequency. When it tries to, because this is a very unstable state, it tries to come back to the ground state. It releases photon as it does that because it has no energy. It needs to release that if it has to come back to ground state. But now the energy it releases is less than the energy it had absorbed to go to the excited state. And so what you find is that there is a difference between the energy it had absorbed, this lambda, and the energy that it is releasing, this lambda. So the excitation wavelength is always shorter than the energy released wavelength. Or in other words, if you look at the frequency, then this frequency was higher. So you had a higher frequency when the energy was absorbed, but when the energy is released, you have a lower frequency release. So the emission is always at a lower frequency. And what happens to the excess energy that it still has? This energy is released by some vibrational motions so that it can slowly come back to ultimately the ground state. So this occurs in a very, very short time. You can delay this coming back to the ground state if instead of going directly to the excited state and coming back to the ground state, it goes into something called a metastable state. There the electron can remain for much longer times, nearly seconds, and then it can slowly come back to the ground state. Anyway, we are not going to talk about anything about the metastable state. Let's go back to fluorescence. So, Ratnesh, fluorescence is a very specific phenomenon. You cannot excite all fluorophores with all kinds of wavelengths. It depends on the electronic structure of the molecule, what kind of frequency of light it would absorb. There's something, somebody has written something?
Yes, ma'am. Like I have found this on Google. Like
Perfect.
it is saying like
घर पे ये
yes, ma'am.
पढ़ लेना, ठीक है, अच्छी तरह से पढ़ लो ये. So not all molecule will absorb all wavelengths. It depends on the electronic structure or the structure of the molecule, the electronic state of the entire molecule, and not just one single atom. It's the entire molecule, uska complete electronic, what should I say, the electronic cloud, whatever you can say, that would determine कि वो कौन सा wavelength, which wavelength it is required to excite the molecule. Once you're able to excite the molecule, the electrons move to an excited state, but the excited state for man is not long-lived. It comes back immediately, almost immediately. There is a lag, for sure there is a lag, but it comes back immediately to the ground state because that ground state is its most stable state. But when it comes back, it doesn't come back to the exact same energy levels. There is a certain, so because it does not come back to the same energy levels, you would find that the energy that is absorbed during excitation of this molecule is always much higher than the energy that is released when the molecule comes back to its ground state. The rest of the, what happens to the rest of the energy difference? There is something called vibrational relaxations that occurs in the electrons that helps it to come back exactly to the ground state. So ultimately, the main thing is that the frequency or the wavelength at which the molecule is excited is different from the wavelength that the molecule emits when it releases fluorescence. So this basic thing is fluorescence, but fluorescence has, you could go on studying fluorescence. There are a lot of things in it. I'm just trying to give it to you in gist. So this is fluorescence. What is key here is that you need a very specific wavelength of light for excitation. And so when the energy is released, the emission wavelengths are longer than the excitation wavelength. And because, again, of the nature of the electronic or the electronic cloud around this molecule, you would see that the emission also has a very specific spectrum. We call it spectrum because you'd see that it has, there is a range of wavelengths through which you will be able to see the emission. And so we always say something called the emission maxima and the excitation maxima. So let's go back here. So you would see that there is a point where you see the highest number of photons being either absorbed or emitted. So we generally classify molecules because they have broad, some of them can have very broad emission ranges, some of them can have narrow emission ranges, so we generally characterize them by looking at this maximum. Okay, so that's about fluorescence. I will start sharing the PowerPoint again. So, slideshow. So now we are now going to talk about total internal fluorescence microscopy. So in total internal fluorescence microscopy, our objective is to see the fluorescence that is emitted by molecules, emitted by say organelles, whatever it is, inside a cell, or by single molecules on the surface of a glass slide. So why is it important for us? Because all these motor proteins that we are talking about, to see their motion, the most important thing is to be able to put them on a glass surface, right? To put microtubules on a glass surface, you label them with a certain fluorophore that would emit certain fluorescence, and then you try to, say, label Dynein with another fluorescent molecule or kinesin with another fluorescent molecule, and then try to see how it moves on the microtubule. So all of this phenomena is near surface phenomena, right? Near surface phenomena. So why can't we do it with plain fluorescence imaging? Why do we have to do it with turf imaging? So in, in, uh, in turf imaging, what is important is that it is going to excite molecules very close to the surface. मतलब bulk solution में अगर कुछ है तो वो excited नहीं होगा. Very close to the surface are the molecules which will be excited. If that is the case, if it can only excite molecules very close to the surface, then all those unwanted fluorescence that may have made it difficult for you to visualize this particular event is actually gone. क्योंकि आप कभी एक माइक्रोट्यूब्यूल डाल के छोड़ नहीं देते, आप उसको वाटर में रखते हैं. Every all of these, the very first day, from the very first lecture I've said that biological motors work in an aqueous medium. तो आपको you have to add water, so you have to have a certain bulk solution in which your microtubules would be on the surface of a glass. So you can have microtubule monomers, microtubules, kinesin motors in that bulk solution, and there would be certainly some of them attached, sticking to the glass slide, and there would be some kinesins or dyneins that are walking on this microtubules, but there will also be some that are floating around on the surface. So if you excite all of these molecules, then all of them will start emitting. Very difficult for you to clearly visualize what is happening on the surface of these microtubules. So to very specifically excite the microtubules, the kinesins, the dyneins, actin, myosin, anything that is very close or literally on the surface of this glass slide, we use turf. Otherwise, we would have excited all the fluorophores, got all the bulk information, and then we would have to use extensive computation to see kaun sa hai, kya hai, and even then the clarity wouldn't be high. So this turf, what it does is it increases something called the signal is to noise ratio. So signal यहाँ पे कहाँ से है? जो microtubules है, जो polymerized हुए हैं on the surface of the glass. So that's giving us the signal. The rest of the solution, whatever it's giving us, is actually for us a noise, and we would like to reduce that, and we would like to see what is happening exactly on the surface. So signal is to noise ratio is very high. It gives us really good clarity, very clear images, crisp images of what is happening on the surface. But it is not that you would only have to see a turf image with purified proteins. You can also do it on cells. So cells में हम क्या देख सकते हैं? Cells में हम देख सकते हैं events which are happening very close to the membrane. So if this is the cell, then you can see that a part of the cell is actually sitting on the glass surface, right? You have this membrane which is literally on the glass surface. And any event that is happening very close to this membrane, or maybe events on the membrane, for example, a membrane protein diffusing on the surface, membrane protein diffusion, yeah, endocytosis. There is an event where a part of the cell membrane invaginates inside and is pinched off and it's taken inside. Yeah, diffusion of proteins on this lipid bilayer can be very easily traced by turf. अगर आप ये normal fluorescence microscopy से करना चाहते हैं, you would have excited all the fluorophores everywhere and not specifically here. Why are you able to specifically excite molecules very close to the surface? Because of the property of the evanescent wave, that it decays exponentially as it goes away from the surface. So very thin, very thin. So how thin? It's between 100 to 200 nanometers. So anything between 100 to 200 nanometers, that small distance, you would be able to excite fluorophores in that range. And why is it given as, say, a range? As I said, as you keep increasing the angle of incidence, you make it more oblique, you see that the evanescent wave falls down even sharply. So closer to the critical angle, so if this is the critical angle, this is the angle of incidence, you increase the, so this has to be more than the critical angle anyway. So as you keep increasing this angle, you would see that the depth of evanescent wave actually keeps decreasing. So you have between 100 to 200 nanometers. So this is turf. Now, how much time do I have? I have 15 minutes, so I think I can do this. So so today I'm going to, यहाँ तक किसी को कोई question है? I've tried to explain it to you in as basic, simple words as possible. But if you have any questions, I can take it, say 2 minutes. No?
Ma'am, here how does the fluorophores bound to these microtubules specifically? Do we use the GFP or?
You can tag them with a fluorophore. You can, so I'm actually going to show you now how to tag a dynein. You can tag it with a fluorescent protein. You can also chemically tag a fluorescent molecule with it. So you could have certain moieties with which, say, so because there are amino groups, you can use simple chemistry, simple organic chemistry to actually chemically attach a fluorophore to the ends of protein. So all of these can be done. You can have both.
But ma'am, in case of like chemically attaching it, won't it be like attached with other proteins also, and not specifically?
Yes. That you cannot control. Until and unless you have a moiety. Okay, चलो next slide पे चलते हैं, आपको समझ में आ जाएगा. So here we have a molecule called dynein. That is what we are studying, right? And what is the question that we had now? So the question that we have to ask so far, what we have seen is dynein का energy कहाँ से आता है? कौन से part of the dynein molecule से dynein को energy मिलता है? अभी हमारा question ये है कि dynein microtubule के ऊपर कैसा चलता है? Because kinesins have a certain walk, right? They can walk one leg, then the other, then the other, then the other. We know how myosins walk. How do dynein walk? So this question was addressed by a number of these publications which came out around this 2000. And here's a specific one that we are going to look at today. And what they did is very interesting. They took a dynein, one dynein molecule, one dynein moiety, and in the gene sequence, they added a certain sequence of an enzyme right into the dynein gene sequence. So this entire dynein sequence does not have the N-terminus. You remove the N-terminus, it's still able to function. But just like in previous paper, वो N-terminus को remove करके GFP डाला था, you remember? Here instead of GFP, you add a very specific enzyme. So a specific enzyme will have, so any enzyme will have a certain substrate, right? Now, what if the substrate is a fluorophore? You got the point? You have, last time उन्होंने क्या किया था? N-terminus को हटा के GFP डाल दिया था. अभी हम GFP नहीं डाल रहे. अभी हम N-terminus को हटा के एक एंजाइम डाल रहे हैं. और इस एंजाइम का सबस्ट्रेट जो है ये फ्लोरेसेंट है. Okay, so and if you now, so why is this, ये इस तरह का अप्रोच क्यों लिया गया है? इस तरह का अप्रोच इसलिए लिया गया है because here it is much more versatile. वहाँ पे अगर आप GFP डालते हैं तो आपके पास एक ही टाइप का एक्साइटेशन एमिशन रेंज होता है. बट अगर आप ये एंजाइम डाल रहे हैं तो आप सबस्ट्रेट को डिजाइन कर सकते हैं in such a way that you can have multiple different excitations and emissions possible. आप बस सबस्ट्रेट का इलेक्ट्रॉनिक स्ट्रक्चर थोड़ा थोड़ा बदलते जाइए, आपको डिफरेंट एक्साइटेशन एमिशन रेंजेस मिलते जाएंगे. So you can actually have multiple kinds of these fluorescent moieties sort of attached to now this dynein. तो उन्होंने वैसा ही किया. And what they did was, now they wanted to see how this dimeric dynein actually works. So आपने N-terminus हटा दिया तो ये dimerization करेगा कैसे? So you can add certain structures to the dynein for it to dimerize. जैसे very simply, there is something called GST, glutathione S transferase, and this GST is known to dimerize very heavily. So that's one way that you can make dyneins dimerize. You add certain moieties to it that themselves dimerize. जैसे coiled coils होते हैं. If you add coiled coils, they will coil. If you add coil domains, the two structures would coil against each other, right? यहाँ पे वैसा ना करके you add certain structures that themselves like to form dimers, either GST, or they have also used DNA structures which can which can complement each other, form these helices, and therefore the two dyneins can come together. So अलग से एक डाइनिन प्रिपरेशन किया जहां पे एक टाइप का फ्लोरोफोर ऐड किया, अलग से दूसरा डाइनिन का प्रिपरेशन किया जिसमें एक दूसरा फ्लोरोफोर ऐड किया गया, दोनों को मिक्स किया गया such that you end up with molecules which have two different fluorophores attached to this dimeric dynein. सुनने में आसान है, but the problem is ये करना थोड़ा सा ट्रिकी है. अगर आप GST डालते हैं then these two of these same molecules could combine, but if you add the DNA very specifically, that can ensure that आपका जो complementary strand है वो इससे ही आ रहा है ना कि इससे. So you can then ensure that you have these two dyneins with two different fluorophores attached that come together, dimerize, to give you a functional dimeric dynein molecule. तो रत्नेश, आपका क्वेश्चन आंसर्ड है इसमें?
Yes, ma'am. Yes.
Okay. So they let this dynein molecule walk on microtubules which are present on the surface of a glass slide. Now this entire motion has to be captured by turf microscopy. इसीलिए मैंने आपको टर्फ एक्सप्लेन किया है, क्योंकि अगर आप नॉर्मल माइक्रोस्कोपी से ये करना चाहेंगे, you will not be able to get this crisp images to do the rest of the analysis because the noise from the background would be very high. You will be exciting all fluorophores. Whereas turf में आप very specifically, because of evanescent wave, बस उतने ही fluorophores को excite कर पा रहे हैं जो एकदम आपके water and glass boundary पे है. So just at the boundary, just at this interface, जो molecules है उनको ही आप excite कर पा रहे हैं. So ideally the fluorophores that are present on the dynein that is walking on the microtubule would be excited. What is the next thing they did? Now, because you're very specifically able to excite these fluorophores, if you use a very good resolution, so there are a few things here. You have to use something called a high numerical aperture lens. I'll not go into the details, let's forget that. If you're, let's simplify this. You have used optics ताकि आपको very clear, crisp image मिले. आप exactly देख पा रहे हैं कि आपका molecule यहाँ है, फिर यहाँ जा रहा है, फिर यहाँ जा रहा है. Exactly like the videos we have seen previously, right? We have seen kinesins zipping past microtubules. Dynein का भी हमने दिखाया है. They're moving on microtubules. So उस तरह का अगर आपको very clear, crisp image मिलता है, तो आप उस image से वो जो आपको फ्लोरोफोर आपको एक स्प्रेडेड आउट रीजन जैसा दिखेगा. अगर आप उस स्प्रेडेड आउट रीजन का एकदम सेंटर और द सेंट्रॉइड अगर आप पोजीशन लें and you keep following that centroid, it will give you the exact position of a single fluorophore. तो इस तरह से उन्होंने इमेज किया है. And then, so they have now the dynein molecule that has the two fluorophores attached to it, right? The two dynein molecules, दोनों में दोनों different type के fluorophores हैं. And now they are looking at this with turf microscopy as it walks on the microtubule. तो idea क्या है? How does the dynein walk? Does it walk like this? This is kinesin walking, right? एक पैर फिर दूसरा पैर फिर दूसरा पैर, या फिर does it walk like this? This is called inchworm. Inchworm देखा है, ऐसे move करता है. Worm, just worm like it grows on vegetables. So it can walk like a kinesin. It can walk like this. So what is the pattern of walking? तो अगर suppose दो conditions हम पहले सोच लें. एक पैर को हम सोचते हैं purple और एक को red. If I think that it walks like a kinesin, then पहले purple आया, फिर red आया, फिर purple आया, फिर red आया. If it goes like an inchworm, purple, red. Purple, red. Red is never crossing the purple. Now let's look at this slide. So here again you see these two fluorophores, इनका जो position है ये real time में उन्होंने इसको mark किया हुआ है. Very simply, real time में जो position है दोनों डायनीन्स का उनको वो देखते जा रहे हैं, mark करते जा रहे हैं. तो अगर आप ये वाला image पहले देखें, you see this is the red, one of the Dynein motor. Blue is the other Dynein motor. So if it was kinesin, you would see that once the red is taking over blue, then at another time the blue is taking over red, because एक हाथ पहले फिर दूसरा हाथ. तो यहाँ भी ऐसे दिख रहा है कभी कभी, red is coming forward, then the blue is coming forward. It's like a step, then red. But कभी कभी ऐसा नहीं है. कभी कभी ऐसा भी है कि the blue comes forward, then the red, then the blue comes forward again, then the red. This is with time. So it's both. So what appears is that dyneins, unlike kinesins, do not have a very fixed motion. ऐसा नहीं है कि पहले एक पैर फिर दूसरा पैर फिर एक पैर फिर दूसरा पैर. It could be a mix. You could have it walking like this. In that case, you would have first the blue line coming forward, which is, you see that the blue fluorescence has moved forward with time. So what are we looking at? We are looking at the position of the fluorophore with time. With time, blue पहले आगे बढ़ गया है, red पीछे है, अभी red blue से भी आगे बढ़ गया है. This movement blue से भी आगे बढ़ गया है. यही हम यहाँ देख रहे हैं कि पहले red ऊपर था, अभी blue red के ऊपर आ गया है. This is in nanometers. Distance traveled, this is with time. So part of it could move this way, and it could also move in this way, that blue आगे बढ़ा है, फिर red बढ़ा है, but ये blue से आगे नहीं बढ़ा, which means that यहाँ पे blue था, यहाँ पे red था, red just about catches up with blue, but doesn't cross it. Then the blue moves forward, the red just about catches up with. So Dynein could walk like this. It can have both of these kind of motions. आज के लिए I will stop it right here. I will continue with this because this is a very interesting way to see or analyze how molecular motors walk. And this technique has been used for both kinesins and dynein to understand a lot of things. So जैसे हमने देखा था कि optical trapping से आपको पता चल जाएगा कि step size क्या है. You can understand that अगर आप directly देख पा रहे हैं कि एक with time, with such precision कि the fluorophore has moved here, then you're literally looking at something called step size, right? कितना move किया है with time, you can actually see that. So this technique will tell you how it is moving on microtubule, what is the step size. All of these information can be obtained from this technique. So we will continue this on Friday. And Friday, I would like to give you a brief flavor of something different because मैं भी आपको एक ही चीज पढ़ा के थोड़ा ऊब गई हूँ. So what we will do in the next class is to look at just videos of how bacteria move and very unique things about bacterial movement, and we'll also connect it with a little bit of this microtubule movements. But we are going to continue this part of Dynein in the next lecture. So, do you have any questions? Sharon? Any questions? Maybe आज बहुत खराब पढ़ा है, कोई question ही नहीं है.
No, ma'am. Ma'am, please send that assignment and also the paper.
I'm very sorry I could not do it yesterday, but I will send your assignment and I will upload that paper today itself. जो
Ma'am,
last
Okay,
time
ma'am.
will upload it today itself.
ma'am, what would be the deadline for the assignment?
You will have at least 10 days time to submit it. Do not worry.
Okay, thank you so much.
Okay. Okay, then we will meet again on Friday, and I will take on with the Dynein, and we will look a little bit on the motion of bacteria. Not literally the flagella, we'll come to that later, but just at the motion of bacteria and what we can do with the motion of bacteria. All right. Thank you, students. Thank you so much, and see you on Friday.
Thank you, ma'am. Thank you.
Thank you, ma'am.